I must at the beginning already discount some of the expectations. Maybe the title, that rough summary that I gave Rahul ended up in this rather grand title. But what I'm really up to is to give you some examples of this curious relationship between physics, some areas of biology. In particular, I'm going to be interested in evolution and information theory. And as we have already heard from Eric, they are sort of different flavors, but have all a common grounding. But on the other hand, there are important differences in what is important to the different areas, what they emphasize. And this kind of interplay is what I want to discuss. So I start with what at the moment is close to our heart in our research, with the first part of the title, molecular evolution. And this is a picture of Darwin, and this is what made him famous. He gave an interpretation of the dynamics that generates what we see as the outcome of evolution. And here we have already one important aspect that goes beyond information theory, both in molecular evolution, evolution as a whole, and in physics, is that we have dynamical laws. So we have a whole body of models that we build that in the first place, as we'll see, have nothing to do with statistics and have nothing to do with information theory, and we glue these models together with probabilistic concepts. And that is part of the excitement, but also part of the conceptual difficulties. Because if you open a physics book, or if you open a book on population genetics, these intrinsic concepts, the dynamical concepts, and the statistical concepts, they are all intertwined. And it's a kind of non-trivial thing to try to disentangle them, but maybe that's sort of in the venue of this workshop, it's maybe a good place to discuss some of this. So here is what Darwin said about this relationship between dynamics and statistics. He said, well, this, what we see, these differences, they are the outcome of a dynamical process that has one stochastic element. The stochastic element is what creates these differences in the population in the first place. And it has a deterministic element. This is natural selection operating. Thank you. Operating within the population and amplifying the frequencies within the population, the proportions, the fractions of individuals with a certain feature that we see, and in such a way that in the end, on different environmental conditions, we see a different outcome of this coupled process. So here, a famous example of Darwin's finches. He saw all sorts of different beak forms, and they are adapted to the kind of food intake that these different birds have in different habitats. But they're not formed in a sort of finalistic way, but they are the outcome of this combined process of a stochastic generation of variants, and then natural selection acting on this variance, and picking out then, and as a population of people, fixing that variant that is most adapted. That's this process of adaptive evolution, and here you have a stochastic component, and here you have the natural selection as a deterministic component. Now, what makes this thing exciting is, of course, today, as we heard in Eric's talk, we see this process not only at this macroscopic level, but we see the genetic basis in this, and we can try to link what we see in genomes to this outcome of this process at the macroscopic level. You see already sort of that we need to think in terms of these probabilistic and information-theoretic concepts in the context of a dynamical process that generates this as an outcome. So here is my one-slide introduction to population genetics. And I apologize to all the aficionados, but of course I will omit many things. So what's the subject of this? Well, the subject is about distributions in a population. So we think of different types in a population. So this could be something macroscopic, like this beak length, or it could be the fraction of the population that have at position number 512 in their genome, letter A, G, C, or T, in which case there would be just, at the single position of the genome, there would be 4 possibilities. So we would have 4 probabilities, or 4 frequencies, fractions of individuals observed in the population that would have to sum up to the total number of individuals in that population, or if you divide by that number, it would have to sum up to 1. So, but now, so we have to specify what are the dynamics that change these probabilities over time. Well, so here I have, for example, 4 such types that are equally distributed in the population at this imaginary initial time, and there are different mechanisms by which these frequencies change. One of them is this deterministic aspect. We think that number 4, maybe individuals of number 4, have a systematically larger effective growth rate. Essentially, difference between birth rate and death rate, but averaged over many generations. Because as we will see at the instantaneously, it's not fitness is not a good question to ask at the level of a single individual. But if we average over large numbers of individuals and over sufficient intervals of time, we can ask whether, say, all the individuals that have letter A at this position of the genome, they might have a slight reproductive advantage, and hence their fraction in the population would tend to go up in time. So, but of course, if you think about fractions in a population, this is a null-sum game. So the total sum is always 1. The increase in fraction of one of these types is going to be the cost of another type. So this will be some time later the outcome of this deterministic process, and you'll see, or you see immediately, that if it's as simple as that, if it's just different growth rates for different types, the outcome of this process is completely boring. It's just the one with the highest growth rate will win. We don't see any variation in the population. We don't see what we see in natural population. We would see a single thing. And everything else would have died out. And since this is an exponential, so this goes and this goes very fast. Now, of course, this is not the whole story. There is a random element here. It's, for example, we can have mutations. So individuals that have a letter A, say, or in this class here, they can change into one of these other classes by mutations that happen in their genome. So that's a stochastic element. It turns out mutations in most organisms, and the organism I'm going to talk on in my last lecture on Tuesday morning about is going to be an exception, but they are kind of a very slow process because molecular machinery has developed all these error correction mechanisms that makes this process happen with a very small rate of error. There's a much larger stochastic element in most populations. The dominant stochastic effect is not these mutations, but is the simple fact that even if you have an average of 1.7 children, you don't have 1.7 children. You have 0 or 1 or 2 or 3 children. And that means because the populations are finite, the number of actual observed offspring is different from the expectation value which is, and it differs from this by what physicists would call a finite size effect. And think of putting this on a computer, this process, and the computer caricature of this would be, you have your n individuals, maybe 50 of them are red and 50 of them are green. You randomly draw, then you draw the 100 individuals of the next generation. You randomly assign a father individual to each of them, copying the color of this father. And then it's obvious that, because the fraction in the next generation doesn't have to be exactly 50 to 50, but it could be 48 to 52, or 46 to 54. In other words, there's a diffusion process in this space of frequencies. And that, you think what this would do the long term, it would actually also drive this process to one of the boundaries. At the end of the day, if you had only this diffusion process, we would always also end up with just one type in the population. So these two, this random process and this selective process, have one thing in common. One of them is stochastic, one of them is deterministic, but they would both reduce the variation in the population over long periods of time. But then that's where mutations come in. There is a slow but steady process that creates novelty. And that's the kind of dynamics upon which our probabilistic picture has to be built, and that our information theory has to take somehow into account. So I will not show many equations. I will just show you these equations. I will also show them in a rather sort of summary way to just convince you, this is details are not important at all, that for that kind, what I've described, for these four pictures of the previous slide, people have canonical equations to express that dynamics. And of course, so that would mean these frequencies, and if you think, so this is a very harmless sounding letter, this lowercase x, but it stands for a quantity in an extremely high dimensional space. So if you think, it's okay if you just have, the ones that have the short beak and the ones that have the long beak, then there would be just two different types in a population, and you have two different numbers, and these two different numbers add up to one. If you think of the space of genomic possibilities, then you would have a different component, in extreme case, in the most microscopic case, for each of these genomes. Because there are vastly more genomes than there are members in a population. So most of these numbers are zero, and some of these numbers have non-zero entries. So it's just as in statistical physics, a space that is much, much, much bigger than any population or even any group of populations can sample. But that's what these lever x means for the frequencies in our base space, if we think of it at the genomic level. Well, so it does change. It does change by selection. So there's a deterministic right-hand side of this equation, and it says essentially these changes in these frequencies, which are actually mathematically, you think of them as a vector field in this high-dimensional space. They push one point to a neighboring point, and then there's sort of something like a susceptibility matrix that says how much of a gradient in one direction results in a change in this coordinate x. Well, there's something more. You can change this by mutation. This is actually also some operation acting on these x's. In this case, it's sort of multiplication with a mutation matrix. And then there is something stochastic. There is something that generates probabilities out of that. So we have a noise term, and this stochastic changes, they have one prominent feature, that is that they are, that's the properties of that sort of noise. I've taken here the simplest form of a white noise, but the important point is that it's inversely proportional to the population size n, expressing exactly that fact that if the population were infinite, our mean field calculus would be correct. Deviations from this are just sampling fluctuations in a finite population. So the smaller the population, the more is that randomness. So that's this in a theory. We're going to compare this to physical theories. This is going to be our analogous quantity to, say, the temperature. Yes?
Can this be derived from a master equation, or is it purely phenomenal?
No, this can be derived from a master equation. I've just chosen here this continuous terminology for ease of description. But you can all this, and of course, we have a discrete, basically a discrete space, right? We have the n individuals in a population, and any of these fractions can be any number from 0 over n, 1 over n, 2 over n, up to n over n. And between these states, you can have your jump probabilities in the sense of a master equation. So, now just to flash you what this really means is, of course, we are talking about the diffusion equation, a very high-dimensional space. And here, for a moment, but please forget this immediately, I've just added the coordinate indices of that space. But what can we use it? So, we have made a transition, right? We have introduced something in this dynamics that makes things stochastic. So now we can talk about probabilities. And so we can talk about the probability distribution over these axes. So now we have an ensemble of many populations. So already this space X was a horrendously large space. Now we have a probability distribution over these x's at a given time t, and that is then, so this would be for physicists a Langevin type picture, stochastic evolution equation, and we can convert this Kolmogorov or Fokker-Dumpf equation for this probability distribution. And of course, we have to find exactly the same three terms on the right-hand side. One of them is this diffusion term, which we were just discussing. This is stochastic changes in x due to sampling from generation to generation, due to the finiteness of the generation. This is why we have here this 1 over n in front of it. Then there is the deterministic change. Again, just think of this as a vector field giving a flow in this space of x's. So this would be the non-stochastic element in our description. And then there's a change due to mutations. So in principle, at the level of writing down this equation, this is fairly straightforward. Of course, we haven't learned anything so far beyond formalism, because we now need to fill this with biological content. And it's exactly the same thing as in physics. You don't learn much from just writing down Newton's equation. You need to say what is on the right-hand side, what is the force, what generates the force, and then you can make a definite prediction on what should be this evolution on the left-hand side. And I should say, I think this is one of the big problems in this field, because we have lots of stochasticity at the genomic level, at the molecular level. So here I've just drawn you one of these pictures of a protein interaction network. And people describe these by stochastic models. They describe the dynamics by stochastic models. They describe the outcome by stochastic models. Yet, we have to think about what comes out of this is, say, a fly. And no matter what all these stochasticities do, no matter what all these unknowns do, and no matter whether we think of them as subjective or objective or frequencies, no, at the end, what comes out is a fly. And of these flies, especially at the embryonic stage, they look damn similar. They look so similar, even if they are from species that have diverged 60 million years away. It takes sort of for an experimentalist a number of years to train to see even under the microscope if they come from that species or from the other species. And you and I would probably not be able to tell this. So, so the transition of from these pictures that emphasize the probabilistic parts So this kind of pictures, and also many of you will have seen, where these genes interact, and it looks more like the wiring diagram from your radio, because a radio is also something that should do the same thing every time when you turn it on. And so this tension between these kind of pictures, that's really part of the things that we're trying to understand. And there is another discipline, statistical physics, where we have understood this already a little bit better. As Eric mentioned already, so it's a bit non-trivial, still in statistical physics. Think of the map from reversible and deterministic microscopic dynamics to what we see, at the macroscopic level, and irreversibility that we observe. But at least we have one large parameter at our disposal, and that's these systems are really big. And 10 to the 23, there's a lot in the way of averaging for our system. So these systems here were pretty deterministic, although they are not at the macroscopic scale. The abundances of transcription factor molecules in a cell is of the order of maybe hundreds and thousands, but it's not 10 to the 23. So how this amazing amount of system determinism can come out from this essentially stochastic machinery, that's something that people are, of course, very interested in. So I spent how much time so far? 15 minutes, good. So I plan to basically give you 3 examples. Today I want to dwell on one of them, and then 2 more in the coming lecture. So the first has to do with regulatory evolution, and so it's quite good actually that I can draw already on some things from previous talks. And so, as we said, the difficulty and sort of the thing where we have to fill things with biological or factual contents is what do we put in these kind of dynamic equations. And so, how do we quantify our selection? What acts deterministically on these populations? So, the famous picture that has been coined by Sewell Wright in the 30s is that of a fitness landscape, which says, and you can think of this here, your horizontal coordinate x, or various, as we'll see, various sort of projections out of this, so that you can make this a manageable space. And then there is a scalar function, capital F, which sort of acts as a potential, and so such that the vector field that drives our deterministic change in x, such that this is the gradient of this function. Just like you would think of a potential function in physics. So locally, this is a very simple concept. So if you think of a population that has, here has one, part of the individuals have one value of x and part of the individuals have another value of x, then this is a simple observable. This difference in f is the difference in these growth rates. And that's why this proportions in a population will move into that direction. So again, so this landscape is not a probabilistic concept at all. It's something, the properties of this, namely growth rate differences, you can measure. You put bacteria population in a petri dish, you're clever enough, part of these bacteria which are type 1 are blue, and the others are red because you have put a reporter gene that turns into a fluorescent gene that makes them conveniently colored red or blue, however you like them. And then you see, you start with 50-50, you see 3 days later, and if you see 80-20, then you know that the blue ones had a higher growth rate and the red ones had a lower growth rate. So you can measure these fitness differences, and hence you can measure potential. And the exciting thing is, so this was a subject where a lot of theory flourished over the past decades, but now we can actually measure such landscape, and people have measured this landscape. So for in the context, for example, of single proteins, where you have a definite number of mutation going from one state to the protein, say one that confers a resistance to an antibiotic, to another state which doesn't confer this resistance, and people, then you have a rather restricted space. Then you are getting a countable space. You have maybe just 5 different positions in your genome, and maybe just 2 important letters per position. So then you have 32 different genomic possibilities, and you have, and then you can, for each of them, you can measure this fitness. And here, this is coded in. One of them has all 5 positions at 1. This is here in this experiment. The wild type has 0 in all 5 positions. And then there is another, for each combination of these mutations, you can make that combination of mutations by site-directed putting in that letter. And then you can actually more or less make these pairwise experiments, pairwise competitions. You see these growth rate differences, and you can assemble them into this landscape, and then you can ask questions on the dynamics of this landscape and on the statistics within this landscape.
Just ask a question.
Yes.
So in general, you know, we have a Fokker equation, the S of x, the S of x object can be the gradient of a scalar plus the curl of a vector.
Yeah.
Now in equilibrium, the curl of the vector vanishes by detail balance, etc. But is there any argument for why it should be 0 here?
Well, actually, that's a good point, right? So this S, I've taken here the case where this happens to be a gradient of a scalar function, and you can convince yourself that in many cases this is a reasonable assumption from the biology of this. But it doesn't have to be the case. And there are many known examples where there is a curve, like where frequencies could go around in circles. The most, one of the well-known things is this rock, paper, scissors game, right? And this game is realized in some populations. And then so if there are 2 types, blue, 3 types, blue, green, and red, and if they have this cyclic advantage, then you can get a cyclic dynamics. So there's nothing fundamental that makes this the gradient of a scalar function. Thanks for this remark. So,
there are even, I think, some Russian population genetics results that say if you allow frequency-dependent selection coefficients, you can get any trajectory to achieve frequency integration. There are mathematical results for that.
Yeah, I mean that's as soon as you move. So that's as soon as you move away from this discrete things of fixations and really talk about within this space. Even for neutral evolution, it's not curl-free. So you don't even need selection. Even the neutral thing, if you have different mutation rates, can already create such a curve. Anyway, so here is sort of what I want to take you home from this first lecture. We have talked about this dynamics. We have talked about the stochastic element that gives rise to probabilities, information theory, inference, and everything. And in the context of this dynamics. So what is now the link between these information theoretical concepts and the dynamic concept? And this is embodied in this equation. So if we think of we now think of one type of this dynamics that creates what we would call a prior in Bayesian statistics. And we take this as our dynamics without any selection. So the dynamics which is only governed by mutations and these reproductive fluctuations, but where there is no deterministic changes. If we think that over this process running for a long time will, in our Fokker-Planck equation, lead to an equilibrium distribution over x as a p, and we call this p0. And then we think, well, now we look at the case under selection. We take this as our posterior distribution. Then the relationship between these 2 probability distributions is such that the probability distribution under selection is the neutral distribution multiplied by an exponential, and this exponential is an exponential of the product of the population size and this fitness landscape. So that's something that deserves to be thought about for a moment, because this, so what we have here, we have something which embodies our notion about the deterministic forces that have nothing to do with probabilities, our fitness. And we're saying we now have a way of putting them into the stochastic theory. They turn up also within the framework of our stochastic theory, and they are nothing but the log likelihood score, of the relative log likelihood score of this distribution that we will find in equilibrium under selection and in equilibrium under neutrality. So this concept that we have as our deterministic dynamics, turns up now in this probabilistic relationship between these two probability distributions. We can ask, we'll ask in the last 5 or 10 minutes that we have what we can learn out of this for biology. But this is sort of, this is one of the ways of expressing this link, or this, between our dynamical formulation, but this f of x was this potential, the gradient of which was just our deterministic velocity field for our change in x's, and this now turns up within our probabilistic framework. Yes?
So let's try and understand this without the biology. Say I was taking a course in Markov chains, probability course in Markov chains. I will encounter the Chapman-Kolmogorov equation. I will encounter the starting probabilities, and I will encounter the equilibrium. So I have a starting probability, I have a transition matrix, and I have a equilibrium distribution after the Markov chain runs and runs and runs. I get a result relating to the three quantities that I started. Are you trying to say that that particular result can be interpreted in the context of Bayes' theorem as the prior, the initial probability being the prior, the equilibrium probability being the posterior, and the matrix, transition matrix, which leads you to this as a likelihood?
What I'm saying is something slightly
I wish you said it, because that would be ingenious.
Well, there you are. I'm sorry to disappoint you. What I'm saying is something a bit more simple. Namely, if we have two versions of this dynamic, right? So then we go to the equilibrium. The equilibrium state will be insensitive to the starting probabilities.
Yeah.
So hence the starting probabilities will drop out of this problem. But if we now take this dynamics in two different versions, one of them is the one without any potential f of x, and the other is the one with a potential f of x. Then we get two different equilibrium states because we got two different transition sets, matrices. And the relationship between these two different equilibria is given by this relationship. We flash the next equation just to say the same thing, a slightly different way. What this means is that the Kullback-Leibler distance, or relative entropy, between these two distributions, which is nothing but the expectation value of this log here, this e to the nf, under this probability distribution, this equilibrium distribution of the full process, is equal to the expectation value of to the average fitness under this distribution P.
Okay, so you may not have said it, and you may not have meant it, but I think I see something completely different which didn't occur to me. Is take a Markov chain, take a course in probability Markov chain without any application in mind, and you get these kind of results. And can you look at these results in the context of Bayes' law, and of course the distance between the posterior, the equilibrium and the prior can be, the two distributions can be evaluated in terms of the Kullback-Leibler distance. So maybe this is not what you have in mind, but at least you're sparking an idea in my mind.
Good. So let me take the last 5 minutes.
Normalized.
that these are both normalized distributions, right? Which implies a given additive normalization of this f.
Okay.
But as we know, all the gradients of this f, this is just like in physics, are completely insensitive of whether we add +5 or -5.
Okay.
By the way, what is capital F of x in the R context?
Capital F is this potential function whose gradients generate the deterministic dynamics, the selective dynamics. So what this means is that we have now this information theoretical aspect of this relative entropy, and this measures something that is a property of our, in this case, evolutionary dynamics, namely the property that Darwin emphasized, that there are fitness differences, which is, from its very definition, not at all a probabilistic concept. So, let me give you one example for how such relationships can actually be put to use. So we have now sort of just imported one of these concepts of our dynamical theory, namely F, into our probabilistic. Can we make use of this? This contact between these two ways of thinking about it, the dynamical and the probabilistic, that have to come together in our stochastic dynamical theory, can we make use of this? And so I'm going to take one example, which you know already by now, so some of the basics, because it popped up in several talks, is transcriptional regulation. So here is the basic process. And so the reason why I chose this example, the reason why we got interested in this example, is that one of these binding sites, you just heard in Uri's talk, this is not a very long stretch of DNA. It's maybe 6, maybe 8, in some bacterial genomes, maybe typical length of 10 base pairs. And this is this site where a transcription factor can land on and bind. And there is a biophysical aspect to this. It has another absolutely non-stochastic aspect of this. Given a sequence of these 10 letters from A1 to AK, where K is maybe of the order of 10, then there's a definite sequence-dependent binding energy. You can measure this nowadays in high-throughput experiments in microfluidic devices. You can measure this for many sequence states. And so you get a list, sequence energy. So you can also actually estimate this bioinformatically. And this, if you take these position-specific weight matrices that Eric mentioned in his talk, they become an estimate of these energy entries for reasons that are quite simple intuitively, not so simple if one wants it at a quantitative basis, but it's quite clear that a letter where binding is more likely energetically is also a letter that you should see more often in the actual ensemble of binding sites. So we have now these short genomes of 10 base pairs, but we have a little bit more. We have also a single real valued number that summarizes these sequence states for us. Because as we read in Uri's talks, like it can be this 4 to the power 10 dimensional sequence space that can still be quite high dimensional to evaluate things. And it can still be quite a nuisance if you want to fill a histogram in this space with 4 to the 10 entries. So having a single valued axis as instead of this sequence space. So this harmless letter A here is of course these 10 letters A1 to AK, and that can still be quite big. So I'm going to finish this up here now and continue on tomorrow with this. So we have now the sequence space, we have a biophysical variable, and we're going to make use of it as a molecular phenotype, as something to quantify our dynamics and our information theoretical inference. This is where we're going to continue. Thanks a lot.
Any questions?
carry the second part of your formalism through if you have those circulating points, because then the stationary probability distribution is no longer exponential.
Exactly.
Yeah.
So this, the fact that you can relate the equilibrium distribution, that there is even an equilibrium in the first place. That's of course, that's a basic result. That's nothing specific to evolutionary theory, right? So if you open a book on Fokker-Planck equations, then you will get the statement that if you have sort of an equilibrium, and then this equilibrium is in the simplest case just the exponential of this force field of the equation. Now here was a slight variant because we had these two equilibria, so all our statements about this one equilibrium was like a relationship between these two, this prior equilibrium and the other equilibrium, but basically that's just what, so that there is two assumptions. First of all, that this force field comes from a scalar potential, and second, that this neutral dynamics is not generating a current. That this leads to a nice equilibrium T0. Both of them are not valid in general.
Yeah, so from a computer science point of view, there's been a lot of interest in genetic algorithms, evolutionary algorithms. It's kind of interesting to see this perspective that you're really trying to go into the data and trying to use this kind of model to understand the data. So I was wondering if you had any thoughts about the sort of bigger picture where, in the case of computer science, they're using this as a way to do stochastic search for optimal solutions, right? So do you have any insights as to, is there some sort of optimization going on that this particular equation is trying to get towards, like a local minimum?
Well, you know, it means that real systems do also some search for optimality, because this is, of course, this is a great question, because we know, of course, that this equilibrium, this is satisfying some optimality principle. It says that this distribution maximizes the expectation value of f at a given value of the entropy. So it makes the, for all the distributions of a given entropy, the one is exponential, has an extremal probability, is the one that maximizes the fitness. This is the one that we have put here as a Lagrange multiplier in this exponential. That's why it satisfies that extremal probability. But of course, this comes multiplied here with this n, which represents the stochasticity in our system. So there is this interplay between high f and the randomizing power of population size, and this interplay is what we're going to see tomorrow in the actual biology. So maybe the answer to your question comes tomorrow: in how far does the actual system actually manage to do some optimization, and how far are the optimization well, I shouldn't say efforts, it sounds too anthropomorphic, but this deterministic part of the time is just wiped out by noise. And so the answer is, sometimes it manages, and sometimes it is wiped out by noise.
One more further questions for the session. Thanks, Michael.